help remind me anytime we meet in Zoom to record it so that anybody who's not able to come can go back and watch it. I always forget to do that. But that's not a big deal because anybody who's not here is in lecture and we've started to record. You guys see the recording in the upper left-hand corner?
Yes.
Okay, great. I'm never sure if you see the same thing that I see. All right. So, I did go into the PowerPoints and did a little bit of tweaking of some of the slides. Not much at all. I might have like actually eliminated a couple slides. And by that I mean if I have like 3 slides, one transitions into another, transitions into another, I just keep adding, you know, bullets. You know what I'm saying? Mm-hmm. I might just have eliminated 2 of the 3 and just given you the entire slide with all of the bullets. That's what I mean by deleted a slide or 2. So what I was thinking we might try to do, and I think I emailed this to you. My initial intention when I first started thinking about how we were going to do this online stuff for actually both my A&P and micro classes was to do PowerPoint voiceovers. And I've actually done that for the A&P class a little bit. But what I'm finding is that it's taking an enormous amount of my time to do a little short snippet. You know, for example, in A&P we're doing the muscular system now. You guys remember when you took A&P 1, we talked about neuromuscular junction and the sliding filament theory and all that stuff. So my initial thinking was I was going to do a big long voiceover, like an hour-long one. And that was just not a good idea because number 1, that's a huge amount of time to be talking. It takes a huge amount of time to upload and download. And so I decided to start using different smaller snippets. So I did one on neuromuscular junction, I did one on sliding filament theory, blah, blah, blah. And so each are like anywhere from 10 to say 18 minutes long. But my gosh, it just took me hours and hours to get just a couple done. So it's not like I'm unwilling to work on this. I am, but it's just eating my entire life. So what I was gonna try to suggest for you guys is, and I'm gonna do this more of an extent to the A&P group too, is any voiceovers that I might do, I'd like to do in A&P and not do them in micro. I mean, I will if you want me to, but I'd rather try to tape the Zoom meeting and share the PowerPoint slide and have us all kind of walk through those sections of the chapters that you're having some difficulty with. Because in all honesty, some of this stuff I don't need to lecture on. You can read it. It's not difficult. For example, in A&P we're doing respiratory system tomorrow, and you know I can go over the anatomy, but you can read the anatomy. They're going to have more difficult time with the physiology of respiration, so I'll do zooming with them. I'll do some overwriting of the PowerPoint slides. So that's what I was thinking of doing for you guys, is tape the Zoom, ask you to have looked over the chapter or a segment of the chapter, and then we get together and we can share the PowerPoint. We can talk about stuff. I can help clarify questions you might have. I'm not trying to throw you to the wolves. If you feel that way, it's not my intention at all. I'm just trying to make the most efficient use of our time. But I want you to know that I'm here to help you in any way I can. What's your reaction to that? Be honest with me. I want to hear what you think.
Why don't we just do like on Zoom calls, like the normal hours of our class, where you would share your screen of the PowerPoints and just take like we were actually in class, and then have you go through. Is that what you're talking about? Like you would just normally go through the slides like you would as if we were on campus with you?
Yeah, that's what we would do, Emily. I think some of the slides, you know, I wouldn't spend much time talking about because they're very kind of factual or fairly easy to understand. But we can certainly take that time if you want to do the maybe 1:15 to 2:30 time frame or whatever. Sure, we can do that. That's not an issue. I was going to talk to you guys about times, like when's a good time to get together? Do you want to do the regular?
I think like as if we were just still sticking with our same schedule, just to keep
continuity. Sure.
I mean, I just think that would be helpful to like get on and have you teach that way instead of just having to like, I know trying to like read it all and teach ourselves sometimes can be.
Sure.
I think that's a good idea, and I would be fine moving from 1:15 to 2:30 like normal, just going over the slides.
Yeah.
I agree.
Okay.
Me too. Me too.
All right. So Thursday we had said, I think 2 o'clock.
Yeah, I think it did say 2 o'clock in the email.
Okay. So for those that are not here, and there's not a ton, how many people do we have? 12. So there's like seven people missing or something like that. They might be thinking we're starting at 2. So could we meet at 2 on Thursday, and then from that point on, we'll do the normal days and times.
Yeah.
Okay.
I think that works.
Yeah, that sounds good.
All right. Great. Okay. So we'll do that. We'll plan Tuesday and Thursday meetings, 1:15, and I upgraded my Zoom account to an unlimited time. I was using the free version, which limited you to 40 minutes, but I was getting booted off because we went beyond 40 minutes for some of the A&P stuff. So we won't encounter that issue. So yeah, we can certainly, I can share the PowerPoint slides. I was envisioning, you know, having you guys tell me what slides you were having issues with or you wanted me to spend more time on. Again, I'm trying to make the most efficient use of our time, knowing that we may end up chatting back and forth more than we maybe would do in class. So can we get through the normal lecture in 75 minutes? I don't know. I guess I would like to have us focus on those slides that you're having particular issues with, and just see how this goes.
So you want us to review all this, like the section for Thursday beforehand, and then when you show the slides, like, do you want us to like all type in the chat like what ones we're having trouble with first, so that you know which ones to like go by, or like how do you want us? Because there's going to be so many of us, if we all have like a different section that we're confused about, I think that would take up the majority of the time, it would seem. I don't know. I'm just thinking.
Yeah. No, that's a good idea. I'm thinking about how to do this as well. I mean, ideally, in a perfect world, you would have looked over. What chapter is this?
Chapter 9.
9.
Okay, you would have already looked over this part of the chapter that we have not yet gone to. We started mutations, whatever day it was we last met. It seems like a year ago to me. And so this is, of course, where we would have picked up had we been in class. So you can use the PowerPoints to guide your reading, and you should be doing that. For example, this slide, you know, I can read this, or you can read this. I don't know that we need to spend time discussing it really that much. I mean, I'm willing to do that if you want, but when we get to these sorts of topics, this is going to involve a little more explanation, perhaps. So if you can read ahead and you can identify what sections of chapter 9 are really confusing to you and bring those up in the chat, then that's going to help guide the discussion. And odds are you guys are going to have the very similar questions probably. Maybe not, maybe. I'm thinking you probably will, but we'll see. And then that can be sort of the focus of our discussion. So rather than a more regimented slide 102, slide 103, we maybe bounce around a little bit, but we focus on those topics that you are having problems with or have need for clarification on or whatever. That's one approach. You guys can see my PowerPoint, right?
Yep.
Okay. So this is a video I just added from yesterday. It should play okay, but you know, this is something that certainly we can play it and watch it together, but again, is that the most efficient use of our time? I don't know. Maybe, maybe not. But you could have looked at this, let's say, yesterday, theoretically, and then when we meet today, if there are questions about conjugation and what that involves, we talk about it. I mean, some people would like just to be given the information as if it was a lecture. I'm just not sure that that's going to be the best use of our time. I'm not sure we're going to get through everything. I've never done it though, so I'm just kind of hypothesizing here a little bit. Have you looked at the rest of 19 or 9, whatever chapter it is?
Yes.
Are there specific sections here that you want to talk about now, or maybe you need to go home, yeah. Sit where you are and read the rest of 9, and we talk more about it on Thursday. You tell me what you want to do.
I know, like on my, I've always had this problem. I don't know if anybody else has this problem, like with the videos and hyperlinks that you add onto the slides. Like the only way I was ever able to watch them was on a campus computer. Like for some reason, they don't come up on that.
No, they don't.
Yeah, mine too. They don't always come up.
Mm-hmm.
So I don't know if anyone has any like helpful advice on how to watch those at home now, because they don't, I can never get them.
It could be, I don't know either. I'm not a computer IT guy.
So it's not just me, and I know Bella has issues too getting the videos that are linked in the PowerPoints.
When you scan over them, does it bring up like a thing for you to hit control and then hit enter?
Yeah.
But it doesn't load? Is that what you're saying?
No, it won't ever like pop up. It says it's like encrypted or something along those lines.
I see.
I don't know if, yeah, I'm not sure why.
I don't know if your virus software is flagging it as suspect.
Yeah, that's what it usually looks like, but I mean, I have my laptop and another computer, so I guess I could try on both and see if I have any luck on the other one, but.
Well, one thing that I've done in the past is use some of the videos that comes with the book. So the publisher provides the videos, and so I've embedded those within the PowerPoints, and those have played, for the most part, pretty well. Once in a while, I'll have an issue where I can't embed it, and I'm pulling the video from a link on my thumb drive. So it's not necessarily embedded in the PowerPoint, but it's on my thumb drive. And so it might be searching that in class, but when you click on it, because you don't have a thumb drive, you just know where to grab it from. So I'm trying to download as many of these videos that I can find on YouTube or the internet and this should play for you. This particular video should play. It's a YouTube video, so it's hyperlinked. If you have internet, it should connect. So that's about all I can tell you as far as the videos go. I'm trying to find those that are available online, so it reduces the chance of any problems. This one as well. It's a little fuzzy to read. It'll clear up when you play it, but this is a YouTube video. I have this video from the publisher, but if I embed it, I'm worried you're not going to be able to play it. So I thought, well, I'm going to be safe. I'll go on YouTube, see if they have it, and they had it, so I put it in.
I've been following a YouTube guy video. He's a professor, and he's following the same book as you. And he does the video with him talking down in the right-hand corner, and I've found that very helpful to watch because he also has all the videos on there too. His name's Edward, and then his last name starts with a K, and I just go in and I type in what chapter we are on out of our book, and then it pulls it up, and I've watched him for just about every chapter we've gone over.
Would you go into the Blackboard course shell and email that information to all of us?
Yes, I will.
Thank you. That'd be great. I'm sure everyone would appreciate that, Corey, if you could do that. And this is exactly what we should be doing. We should be sharing these links, any supplemental outside stuff that you're finding helpful. Share it. This is we got to do this. We got to work together and help each other. So that's the goal.
So I have another question. This isn't really related to micro, but are we still doing our spring break then? Because we've had this time. I guess we haven't really had it off, but.
So your question is, are we still having class as opposed to having a break?
Yeah.
I don't know about you, but I desperately need a break.
I know, I do too, but I didn't know if you were, like, I know my kids' school break is not technically
Yeah.
a break, I should say.
You know, I'm going to use some of that time to work on this. No, there's no question about it. I'm not going to Peru, although I was going to go to California, so I'm bummed out about having to cancel my trip. But if you guys want to meet that week, I mean, we're off for like from the 1st to the 10th or something like that, right? But if you guys want to meet, any of you want to meet that week, I will certainly be available. I'd be happy to do that.
I don't want to meet, but I will if you want to. I want a break too.
Well, it's more of what do you guys want? I mean, I'm happy to do a Zoom meeting.
I just didn't know if, like, because you were saying that you don't know, like, going over the PowerPoints like we normally would in class on like a Zoom call, like if you're worried that, like, the best way to utilize our time together, like, are you worried that because we haven't really done anything like the past week that we're falling behind, and should we? I don't know. Should we do?
Well, you have the updated lecture and lab schedules, right? So we're gonna try to adhere to that schedule, and it's not a heck of a lot different from the old one. But we've got to get through those various chapters in lecture between now and May 12th or whatever it is. So, you know, you need to use your time as wisely as you can in terms of you know, beginning to read those various chapters that we've got scheduled. So I think we wanted to get through the genetics chapter this week, according to the updated schedule, right? And the next chapter 10 is on genetic engineering, and that particular chapter, I also worked on the PowerPoint of that yesterday. That is a really technical chapter. It's it's a very technical chapter. So I chose certain parts of it to put into the PowerPoint because it's just beyond the scope of this course to get into the detail, the technical aspects of recombinant DNA technology and all that stuff.
So
Go ahead.
will those, I guess while we're on break, will those like updated PowerPoints that you're working on be like available to us, or is it just the ones that were already in there? So like we know if we can follow along with that, just so we know what we're studying, I guess.
All the PowerPoints are listed in Blackboard, right? You know that.
I did know that, but I didn't know if because sometimes you like update them before class and take certain things out. Like, is it going to show up as the updated version or the old version?
When I go in and I do edits, I save as and overwrite the old PowerPoint. It doesn't tell you updated on March 24th at noon. I mean, it would be nice if it did that, but I don't think it does. And I can't, to be quite honest, tell you when I'm going to go into chapter 11 and tweak that. But if I do tweak it, it's not going to be a substantial tweak.
Okay.
It's going to be some minor little edits, if anything. Like I said earlier, if I had a 3-slide combo with, one transitioning to another, and I take the first 2 out and just give you the entire slide, that's an edit, but it's not a substantial change for you.
Okay.
You know what I'm saying?
Mm-hmm.
So my goal is to not do much tweaking, if any, but it's not going to be substantial. I really wouldn't worry about that.
Okay.
I think what you've got to start to do is get into the book, read, try to keep up with the schedule, and for those sections that are causing you grief, make note of it, bring it up during our Zoom, or if you want, you could even, you could, I guess, email me questions, and then when we get together, I'll have some sense of what people are having difficulty with. That's a double-edged sword. When I get 40 emails and then from 2 different classes and trying to keep track of all that, it can be a little challenging, but again, I want to make the best use of our time. Maybe we should just go to the PowerPoint for chapter 9 and just kind of walk through it. And I'm assuming you guys have looked at this. Maybe that's a wrong assumption to make, but some of you said you did. So let me just open up the floor for questions on chapter 9, anything on chapter 9. And don't be afraid to ask. I think we last left off talking about mutations. Right?
Yeah, I think it was causes of mutations.
Right. That's right.
That was way back on the 12th.
Yeah. Mm-hmm.
Long time.
Yep. Right. So we talked about, this was our last slide, I'm pretty sure.
Yes.
Now, for the exam, I'm not expecting you to know all these chemicals and all their individual impacts. If that's what you think you need to memorize, that's not true. I think you should understand that in the case of radiation, we're impacting the DNA of the cell. That could be a bacterial cell, that could be your cell, right? We know we sit out in the sun too long. UV light increases the risk of skin cancer, right? It's affecting the DNA of your cells, your skin cells. And that can result in what we call melanoma, right? Melanocytes, you remember from A&P, are embedded in the deepest layer of the epidermis, and if they begin to divide uncontrollably, it can lead to skin cancer. The same idea applies to prokaryotic cells, bacterial cells. If you zap them with UV or a more powerful form of radiation called ionizing radiation, you can really affect the DNA. You can tear apart the DNA for the cells, and the cells can't replicate and they die. This is why UV is sometimes used in the hospital setting. They have UV lamps they'll bring into a room, and they'll let that lamp shine on the whole room, and it sort of disinfects the room. It doesn't sterilize it, but it disinfects the room. So that's kind of interesting. This is a video, I don't know if it's going to play here, I think it should, that talks about how you can have nitrogenous bases rather than the TACG combo that we know exists in DNA. You can sometimes get a T and an A, or a T and a C, or a G and an A. The mutation can result in an alteration of the bonds that form, these hydrogen bonds between the bases. So you remember we talked about T opposite A, C opposite G in DNA? Well, if you get a mismatch, that's a mutation. That's a change in the DNA, and by definition, it's a mutation. And if that's passed on to the progeny, the cells that are formed, then that mutation is passed on, and it could be a problem for the cell. So these slides talk a little bit about mutations, and again, this is just a definition that says if we're impacting just one point of the DNA, let's say a pair of bases, we call it a point mutation. And sometimes the bases are, a T is substituted with a C, or a G is substituted with an A, or sometimes you can have what they call addition or deletion mutations, where we're adding or deleting a pair of bases in the DNA, and that can have potential ramifications that are discussed here. So again, we could go through and do the lecture like this, or you could have looked ahead and be somewhat familiar with what I'm talking about, and we can kind of jump over this a bit. As I said, I think if we go through the entire time, it's just we're gonna run out of time. How many people have looked over this and are familiar with what this section talks about?
I briefly looked over it.
Okay.
So I think before Thursday, I will more thoroughly look over it.
Yeah, I mean, you need to start doing that. Start reading over the chapter, referring to the PowerPoint. The PowerPoints guide you. I mean, they're pretty much sequentially presented as it is in the chapter. So it should be pretty easy to keep track of where this is in chapter 9, right? What section and so on. So this table talks a little bit about different types of mutations, which again are changes to the DNA. And they use this kind of easy to understand concept of, if you had this sentence, let's say, the big bad dog ate the fat red cat. Each of these letters represents, let's just say, a T, an A, a C, or a G in the DNA. Now I know there's B's and D's and other letters there, but that's the assumption. So if you were to, for example, if you were to have if you were to have changed any of these letters, any of the nitrogenous bases, what that could do, let's just pick the insertion here as a good example. Let's say we insert a B here in place of the D. So this is the original sentence. We alter it by putting a B in. What does that do to the rest of the sentence? Well, obviously it screws it up. Just like if you were to delete the B in bad, take it out. What does that do to the rest of the sentence downstream of the mutation? Well, you can't even read it. It changes it, right? So if these three-letter segments represent codons, right? What does that do to the codon sequence downstream of the insertion mutation? It changes it, right? What it does is it induces what's called a frame shift. Everything is shifted down, in this case, one letter. In the same way that if you delete a letter from here, everything is frame shifted. Everything is altered. Now, if this is the messenger RNA, which codes for the protein, you got to understand transcription and translation. Now, I'm assuming you know all that. If you don't, you better go back and remember it, because this makes no sense to you if you don't understand transcription and translation. And we spent some time talking about that. You've got to understand that. What does that do to the resulting polypeptide that would be translated?
Altered?
What do you think?
It's altered?
It's significantly altered, right? These codon B sequences are so changed, we're going to be bringing in different amino acids than we ordinarily would have, right?
Yeah.
And if you bring in totally different amino acids, are you going to be constructing a totally different protein? Absolutely. And odds are that protein is not going to function like it should have had the mutation not taken place. So that's the gist of this whole slide, is to understand how different mutations can have significant ramifications potentially on the protein being produced down the road, or what if you accidentally introduced a base that in turn gave you a stop codon prematurely? What's that going to do? There's no question as to what that's going to do, right? What is it going to do?
It's going to stop the...
I didn't hear you.
I said it's going to stop like the production.
Yeah, you're not going to be bringing in any more amino acids, are you? Right. And so what would have involved maybe 100 additional amino acids being brought in to make the protein, there's 100 less brought in because you stopped translation prematurely, right? That's what that indicates. So these are significant mutations. Could be. Now, you got to also think about the fact that if you go back into that table here, there are some amino acids that are coded for by more than one codon sequence, right? Phenylalanine will be brought in if either of those two codon sequences are found in the messenger RNA. So if you substitute a C for the U here, this last one, is that going to make any difference? The answer is no, because you're bringing in the same amino acid.
Okay.
So mutations may not have any impact on the resulting polypeptide if you're coding for the same amino acid anyway. Does that make sense? trying to gauge reaction on faces, and I'm not doing very well here. Are you guys totally like confused? Are you totally frustrated? Are you understanding what I'm saying? What's the scoop?
I'm okay. I get it.
Yeah, I'm good.
I understand what you're saying.
Yeah, I'm good too.
Okay. So some mutations can be significant in terms of altering the DNA to a point whereby the protein that would ordinarily be produced just isn't produced. Or the protein could be so altered that it doesn't function the way it's supposed to, and that's not good for the cell. Could the DNA be altered such that there is no impact on the protein? Yes, we just talked about that. But what I think is important to understand, and these next few slides kind of talk about how if there are mutations, the cell has often ways of identifying and fixing mutation. It's really interesting. If there's a mismatch, let's say there's a T opposite C or an A opposite G, when the DNA is replicated, DNA polymerase, in addition to bringing in the new nucleotide during replication, can also proofread what's been replicated. And if it discovers a mismatch, it can snip out the bad guy and put the right guy in. That's what this video will show you. Very cool. And there's other enzymes listed here that can also be very helpful to cells as they identify and repair potential mutations. So this slide talks a little bit more about how some mutations lead to non-functional proteins, how some mutations have no impact at all. But I want you to focus on this second bullet. This is really interesting. Organisms with mutations that are beneficial in their environment can readily adapt, survive, and reproduce. What this is getting at is the idea that mutations don't always have to be a bad thing for a cell. Now, once in a blue moon, a mutation could potentially alter the protein a little bit such that it is, say, more effective in combining with substrate to make product, if it's an enzyme. Right? You all know that enzymes combine with substrates and products are produced. And what's the part of the enzyme that combines with the substrate?
Is it an enzyme? No.
No. What part of the enzyme combines with
site?
the substrate?
Is it
The active site?
The active site, right?
Okay.
That's the three-dimensional part of the protein where the substrate binds. It's like a key fitting into a lock, right?
Yeah.
The lock is the active site of the enzyme. The key is the substrate. So let's pretend that the mutation results in a better lock, if you will. And the result is a more beneficial function to that protein. Well, that can be helpful to the organism in the long run. Not even so much the organism, but the population. And this is getting kind of off the topic a little bit, but the basis for evolution at the micro level, at the DNA level, is all based upon mutations. So in that case, mutations can actually be beneficial to populations down the road. It doesn't happen a lot, but it happens enough that evolution occurs. Okay. Questions? All right, this next section of the chapter talks about ways in which genes can be acquired by organisms. and we're talking about bacteria. How do bacteria sometimes acquire new genes, new DNA? Remember, genes are segments of DNA that typically code for proteins, at least the genes we'll talk about. And so this is introducing 3 mechanisms by which bacteria acquire new genes. Sometimes they can acquire them from other bacteria. That's what's talked about here in conjugation. Again, I guess rather than lecture on this, I just would like you to read over your book. I'll show you the video here because I think does a good job explaining conjugation. Let's see if we can watch this together. Is this going to work?
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. F+ cells carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F-. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid, beginning at the origin of transfer, enters the F- cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F+ and F- cells are mixed together, eventually all of the cells become F+.
So this is an example of conjugation. Where a cell is producing what's referred to as a sex pilus or pilus, across which DNA can be transferred. In this case, we're transferring what's referred to as a plasmid. You might go back into the prokaryotic chapter and they talk about plasmids. They're extra chromosomal pieces of DNA. In other words, besides the DNA you find in a chromosome, many bacteria have these little circular pieces of DNA called plasmids. And in this example, the video just talked about how if a cell has the ability to produce a plasmid, or produce a sex pilus, and transfer this F factor plasmid, that cell is denoted as F+, a little superscript plus here. I don't know if you can see that or not on the diagram. The cell on the right, the green cell, is F-. What does that mean?
It doesn't have the circle.
It doesn't possess the plasmid, correct. And because it doesn't have the F factor plasmid, what can it not do?
Reproduce?
It can't produce the sex pilus. It can reproduce, it can divide by binary fission, but the whole idea here is to understand the ability to form the sex pilus across which DNA can travel. So F- can't transfer plasmids to any other cell. It doesn't have the means to do that. This F+ can, because it possesses this F factor, sometimes thought of as fertility factor. It has the ability to make this little bridge, this conjugation tube, it's called. And in so doing, it replicates its plasmid. Okay, this is DNA. It replicates with the help of enzymes. And this is the replicated DNA moving across, like the video showed. Now the F minus cell becomes what?
F positive.
F plus, because it has the fertility factor. It can dock with another F minus and do the same thing. It can transfer it across. So this is introducing the concept of how bacteria can transfer DNA in the form of the plasmid from one cell to another. This F factor is really getting at the idea here of being able to manufacture this little bridge, or pilus, some people pronounce it pilus. It's a little bridge here between the two. This guy makes it, this guy receives the bridge, and then ultimately receives the plasmid. Does that make sense?
Yes.
So that's called conjugation. You should definitely look that over. What is the impact of that? Well, there are other plasmids in certain bacterial cells called R plasmids. Think of the R as resistance. These R factor plasmids impart protection to the bacteria. They resist the action of many antibiotics. Is that good for the bacteria?
For the bacteria, yes.
Absolutely. If you have R factor plasmids, you can resist the action of amoxicillin, erythromycin, blah, 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 potentially, right? So, you see where I'm going with this? If I'm a bacteria with an R factor plasmid, and I produce a sex pilus, and I merge with you, and you don't have the R factor plasmid, what am I going to do? What could I do?
Infect me.
Well, I'm not going to infect you. I'm going to affect you.
Oh,
What am I going to give you?
The R
R
factor.
factor.
Yeah, I'm going to replicate my R factor plasmid just like I did here with the F factor. And guess what you now have the ability to do?
Resist antibiotics.
Exactly. So we talk about drug resistance and antibiotic and bacteria. This is often how that happens. Yeah. And so not only, well, there's a number of ways that you can resist antibiotics, and that's what this second bullet talks about. Some R plasmids allow the bacteria to make different kinds of toxins or to adhere to host cell membranes. In other words, the increased pathogenicity is the end result. And that's a good thing if you're a bacteria. It's not a good thing if you're a human being or a dog or an animal or a plant, because you can suffer the effects of bacterial infection. Diana,
So-
you're thinking here. I can tell you're ready to say something.
Well, when you talk about the pathogenicity and everything like that, is that similar to what they say like about the COVID or the coronavirus? Because they said, there was an article that said that the people that took ibuprofen actually got sicker than the people that took acetaminophen.
Yeah, I heard something about that in the news recently as well. I have not researched that, so I can't comment on the ibuprofen impact, but I have read it. This is bacteria, which is different from viruses.
Right.
From my understanding, we're not talking about, and I gotta be careful when I say this because I'm not exactly sure. Bacteria aren't transferring pathogenicity to viruses via this process. At least I don't think so. But any microorganism, be it a bacterium or a virus, is always constantly evolving, changing the outer signature. Now remember, we talked about viruses, I don't know, back whatever chapter that was. Those envelope viruses had those spikes. Remember, it was like H2N4, H6N7, and those H's and N's represented the type of proteins on the surface. And so what a virus will do, and COVID-19 is no different than any other cold virus. All cold viruses are coronaviruses. They change their antigenic spikes. So even if we develop a vaccine in 12 months or 6 months, quicker the better, guess what?
it might not work.
Exactly. It might not work. Now, it might be good enough that it'll protect 60% of the public, which is better than 0% of the public, right, that we have now.
Mm-hmm.
But like any vaccine for any pathogen, we're always a step behind the bug because of its antigenic characteristic evolving and changing all the time. And we're getting a little off topic, but
Yeah, I'm
okay.
sorry.
No, it's a great question. That's a great question. Okay, do you have a pretty good sense of conjugation?
Yes.
Okay. The second process is referred to as transformation. Transformation. You know, I'm going to just let you spend some time reading over the book, checking out these PowerPoints, and again, this is not rocket science. This is something I think you can understand if you just read through it, spend some time studying it. I'm not going to lecture on it, unless you know you want me to on Thursday. Let's do that.
Okay.
Okay. The third and last process by which bacteria can gain genetic information is called transduction. Now here, we are using a virus to transmit bacterial DNA from one bacterium to another. This is very interesting. Now, when you read bacteriophage, do you know what that is?
That's the lunar lander, right?
Right, that's right. It's the lunar lander, exactly. And you know that the lunar lander goes through the lytic and lysogenic cycles, right? That it infects a host cell like E. coli, and the E. coli responds by being turned into a viral factory that spews out tons of bacteriophage if and when the cell lyses, right? Same old stuff, nothing new here. Okay, in this example, however, bacteria have benefited, believe it or not, by invasion because the bacteriophage can inadvertently pass on DNA from a cell that was infected to another non-infected, well, I shouldn't say non-infected, but to a new bacterial cell. Now note the blue here. This process occurs between bacteria of the same species. So the use of bacteriophages, let's say, has to occur between E. coli. You can't have, you know, the bacteriophage picking up E. coli DNA and transferring it to staph, or Bacillus megaterium DNA being delivered by the bacteriophage to Proteus vulgaris. Okay, it's got to be E. coli, Proteus to Proteus, Klebsiella to Klebsiella, whatever it might be. Okay. Two different kinds of transduction, and I think there's a video here. We want to just go right to the video. See if this will play. I think it should. Okay, that's specialized transduction. Let's talk about general. I don't think I have a video on general. So in generalized transduction, here's E. coli. Let's pretend this is E. coli. This is one cell of E. coli. Here's the phage, the lunar lander, injects its DNA into E. coli. Note again the phage DNA in red, the E. coli DNA here in the chromosome in blue. The end result, of course, is the cell in essence doing what? How's the cell going to respond? You all know the answer to that, right? The cell is going to be taken over. It's going to be making more viral.
Factory.
I'm sorry?
It's going to be like a viral factory.
Exactly. It's going to make more viral capsids. It's going to make more viral DNA, right? And it's going to be packaged into more bacteriophages, which we see here in the middle, right?
Mm-hmm.
Note the DNA in these three phages. What is it?
Correct.
Red. Phage. Right?
Mhm.
This is more of the same of what originally started the infection, if you will. I guess this is an infection. Invasion, whatever. What about this guy?
The host DNA.
Right. This is blue DNA. So it got incorporated into this phage, or a certain percentage of phage. When I say phage, it's a fancy shortened version of bacteriophage. Same thing, same word. Okay, so phage means bacteriophage, lunar lander. So this particular percentage of phage, well, this is 25% of the shown here, but it could be any number, I guess. This phage is carrying bacterial DNA. And this cell is going to lyse, and out pop all these cells, or all these phages. And what are they going to do? Presumably.
Infect other cells.
Exactly. Infect other what cells?
E. coli.
Bacteria?
E. coli, E. coli, E. coli. Remember, this has to happen between same species.
Same. Okay.
Yeah, don't forget that. So here's E. coli number 2 in green. It's invaded by this phage. What's the phage doing? Injecting its DNA, right?
Yep.
Note it, blue, DNA from the original bacterium. This can be incorporated into the chromosome of E. coli cell number 2. So suddenly its genome, has it been changed? By genome, I mean all the DNA.
No.
Has it been altered?
No.
Who said no?
I did.
Oh, Vera?
Yeah.
Okay, I don't see you. Don't you have a camera?
Sure.
No, okay, that's all right. I'm just razzing you. When you compare this chromosome, Vera, to this chromosome,
Oh.
you're telling me there's been no change.
Well, oh,
There
okay.
she is.
Not that I see it.
Is it hard to see from where you're sitting, maybe?
No.
There's some blue in here, right?
Yeah.
So this DNA has been inserted into the chromosome. This piece of DNA is actually becomes part of the chromosome.
Mhm.
So it has been changed. Does that make sense?
Yeah.
Now, when we say this has been changed, can this cell do something different after having been infected versus before it got infected? The answer is yes. What can it do, presumably? Well, it depends upon what genes were in that blue. If those genes allowed that cell to do something that made it more pathogenic or protected it in some way from antibiotics, that could be beneficial to the cell, could it not?
Okay.
Sure. So again, this is just another example of how this bacteria can acquire genes from other bacteria, but who's the taxi driver here?
The bacteriophage.
The phage, right. The phage is the vehicle without which this could never happen. So we think of phage up to this point as being bad to the bacteria or for the population of E. coli, and it certainly could be. But do you see how this could potentially be beneficial if this new set of genes made this cell better able to survive in an environment with antibiotic or whatever it could be? You know, it depends on what's introduced. It could be significant to the cell in a helpful kind of way. Questions on that? Okay, let's look at a video on the second type of transduction, again employing a virus or a bacteriophage virus. Only this is called specialized versus general or generalized. And this is going to be talking about how a specific part of the donor bacterium is incorporated into the second bacterial cell. This is a more, as it mentions, a generalized process. Here we're transferring specific pieces of DNA. Let's see if this plays.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example, the gal gene, near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell, and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Okay, so this is very, very similar to the previous video. Come on, stop here. Only here, the key is transferring a special piece of DNA from the first cell to the second cell. Now, I can't speak to how that process occurs in terms of specificity or getting into a lot of detail, and it's beyond the scope of this course. But the key takeaway is a specialized segment of DNA from the original cell, E. coli cell 1, to E. coli cell 2. A specialized piece. So this slide simply reviews in one table the 3 different mechanisms by which material can pass, genetic material can pass from one bacterium to another. It's a great summary slide. So, spend some time checking that out. This would be a good essay question for a test. I'll just stop, ask, are there questions? Okay. This next topic relates to a phenomenon referred to as transposons. These are sometimes called jumping genes. This was first discovered by a woman by the name of Barbara McClintock, or McClintock, I think is her name, back in the 1950s. She worked on Indian corn, or maize with the colored kernels. When she first proposed this concept of jumping genes, which was the cause of why in, say, a cob of Indian corn, some of the kernels were red, some were yellow, some were striped, some were dotted. You know what I'm talking about. You've seen Indian corn.
Yeah.
All different interesting colors and formations of the kernels of a corn cob. She explained it as those different kernels of the corn contain those different colors because different genes were being expressed that resulted in different colors forming or different shapes of the kernels. There's lots of different phenotypes for corn kernels, believe it or not. And when she first proposed this and wrote about it, she was chastised up one and down the other by her colleagues. I mean, she was just lambasted. But to her credit, she stuck to her guns. She won a Nobel Prize for her work on this. So it just goes to show people tell you you're wrong and you believe in what you're doing, stick to your guns because you might be right. But anyway, it's really interesting. So I would ask you to just watch this video and check out that section on transposons. So the last part of chapter 9 talks specifically about animal viruses. And actually, this is not a whole lot of new stuff. You already have a sense of how viruses work. But we're going to talk just briefly about DNA viruses. And if you remember, in the case of a DNA virus, the DNA of the virus eventually has to make its way into the nucleus of the host cell. So here's the viral DNA that's being brought in through the cell nuclear envelope of the cell. What's the cell going to do? Well, it's going to transcribe some of that DNA. All right, so here's our viral messenger RNA. It's going to leave the nucleus, go out into the cytoplasm, where the cell will utilize its ribosomes and its transfer RNA and basically make viral proteins, which come back in, and these are going to be used to assemble the viruses, viral particles, right? The protein capsids. But what else does the virus capsids, what do they need? In addition to outer protein, what else do they need in order to make more virus? You need viral DNA, right? And so this is describing how the cell not only will use the viral DNA to make the messenger RNA, to make the protein, but we also replicate the DNA so we can pack that back into the proteins and make our mature virus. That's the whole goal here, right, of the host cell, to make more virus. Mm-hmm. So this is nothing new. We've talked about this. You should note, however, that some of this replicated viral DNA, what's going on here? Well, it says, right, it's inserting itself into the host cell DNA. That's interesting. That's something I don't think we've talked a whole lot about, have we? We've always talked about how the viral DNA is the instruction manual to the host cell to make more virus. So this is bringing up an additional kind of topic. And so this introduces the concept of how viral DNA, when introduced into a host cell, has the potential to alter the DNA of the host cell to such an extent that it becomes a cancerous cell. And I'll give you some examples here of some viruses that can induce different kinds of diseases, hepatitis, some of the herpes simplex viruses, papilloma viruses. These are the ones, right, that we talked about recently that we're having our kids being inoculated against so they don't get cervical cancer, right? You've heard of this. Because we know that when we're exposed, some people exposed to certain viruses, the result can be, down the road, cancer. And the reason for that is we are altering the DNA of the host cell, and that host cell becomes a cancer cell. It's a mutation. It's a change in the DNA, an introduction, really, in this case of viral DNA into the genome of the host cell. So these oncogenic viruses are those that can initiate cancer. That's what that means. Onco means cancer, right? Oncologists are cancer specialists. So this cell that has potentially acquired this DNA from the virus and maybe potentially turned it into a cancer cell, we talk about that transformation of the host cell. And you might say, well, what causes that cell to become cancerous? Well, you got to think back, I guess, to ANP. And remember, at least we talked about it in our course, cancer often can result from the activation of oncogenes, right? Or the suppression of tumor suppressor genes. So sometimes the uncontrolled cell division that we connect with cancer is due to certain genes being turned on or turned off, or in this case, we're inserting genes that become turned on, and that leads to cancer. This last slide is talking about another group of viruses called the RNA viruses. Now you remember back in the virus chapter, there's DNA viruses, RNA viruses, right? Well, this particular group of RNA viruses are referred to as retroviruses. AIDS is a good example of a retrovirus. You've all heard of AIDS. How does AIDS cause problems? Well, does anybody know what host cell the AIDS virus infects?
RNA.
Your white blood cells.
What specific type of white cell? You're right.
Your T.
I'm sorry?
Lymphocytes.
Well, leukocytes are white cells. I can tell that some of you are like, oh, I remember hearing about this, you know.
The T
I can
cells.
see you're thinking about this.
The T cells.
Cells.
I'm sorry?
Your T
T
cells.
cells.
T cells, yeah, they are T lymphocytes, right?
Mhm.
Specifically, helper T cells. Yeah. And so let's pretend this is AIDS virus. It's entering, of course, here a helper T cell. We see it uncoated. Well, we see the outer envelope remaining on the surface, I guess. And then we uncoat the capsid. And here comes our viral RNA. Remember, this is an RNA virus. So what does the host cell do? What does the T cell, helper T cell, do in response to invasion by AIDS virus? Well, it, again, uncoats. Out comes the viral RNA. And look what it's going to do. It's going to utilize an enzyme that actually the virus brings with it, called reverse transcriptase. How do I know that's an enzyme? Because it says that. No, you know why it is.
That's an ASE.
You're right. And remember transcription. What was transcription? Tell me in a sentence transcription. Define it.
When a template strand of a nucleic acid is used to like make a copy of the nucleic acid.
Oh.
Or like the template strand of a DNA is used to copy the DNA.
No. You're not copying DNA. You're not replicating DNA. You're copying it, but what are you making?
RNA.
Like
Right. You're making messenger RNA. And that's what you were thinking, Vera. I know. I can tell by the look in your face. Don't confuse DNA replication with transcription. That's the point I want to make. And I don't know if you were or not, but in transcription, you're right. We're using one of the two template strands of DNA to make messenger RNA. Okay. What does reverse transcriptase do? It reverses transcription. Right? We're taking messenger RNA, or in this case, we'll call it viral RNA, and we are making double-stranded DNA from it. The host cell does that. Okay? With the help of this enzyme. You could not do this without reverse transcriptase. The virus brings aboard that enzyme. The cell responds by reversing transcription, taking RNA and making DNA from it. So that's what's shown here in blue. This is we call it proviral DNA. It has to be inserted now into the chromosome. And now it's viral DNA as opposed to pre-inserted proviral. So it gets inserted into the DNA of the host cell. And what does the host cell do? Well, it transcribes that viral DNA into messenger RNA, and that has the instruction booklet to allow for the formation of proteins, viral proteins, right?
Mhm.
And we also can make viral RNA, package that inside the protein, and there goes our new AIDS virus to infect other T helper cells. The other very interesting part of this is mentioned here in the pink box. The provirus of HIV can stay dormant in cells for years. So what we're saying is that this cell can sit around for a long time with proviral DNA inserted into its genome. And down the road, something could trigger, the release of virus. What in essence this does is it puts the patient at a very compromised immune status. Because if you remember from A&P, and we'll review this in another couple of weeks in micro, the immune response depends on T helper cells. And the analogy that I often make, and maybe I mentioned this to you guys, I just can't remember. Think of a bar stool, right? You got 3 legs that holds that bar stool up. Imagine taking a saw and cutting one of the 3 legs off. You're going to fall over, right? That's what happens with the immune system. You take out one of the 3 legs, in other words, the T helper cells, and they can't fight the invasion because they're too damn busy making HIV. Yeah. Okay. That finishes up PowerPoint. So, this was pretty similar to what a lecture would have been, I guess.
Right, and I was just going to mention, I do like going over it as though we were in class.
We can try to do that, but I will simply say that if we find that we're not getting through the information or the material in the schedule, we're going to have to do something differently.
Right, I understand.
In other words, on Thursday, we're scheduled to look at chapter 10. I'm not sure in an hour and a half or 2 hours, we're going to be able to do that without you having looked that over ahead of time.
Right.
But I fully get what you're saying, and I would have the same reaction if I were sitting where you are sitting. I'm just going to suggest that while we can certainly do that, I have to be mindful of the time. And so I'm putting a little bit of pressure on you to do some reading maybe ahead of time so that we can get through the material. I don't want to rush through it because that makes everybody anxious, worse than we already are. I get that. This is a totally different platform we're trying to do here.
It is, but like you said, we'll all work together. We'll figure it out.
It's not optimal, but it's kind of the way it is. And I guess that doesn't also preclude the idea that whatever we don't get through in 10 on Thursday, that we couldn't spend some time over the break. Although I don't want you to feel like you need to necessarily come to class during your vacation. But I'm willing to meet if you want to meet. And we can tape this. Hopefully this is going to be taped and I can post it on Blackboard. Let's just open the floor for comments about how this last hour and a half went. Anybody want to offer commentary?
I also like how we went through the slides, but could we go at our normal class time?
Yeah, absolutely.
I also liked going through the slides, but the normal class time would be good as well.
Yeah. Let's do that. I think for Thursday, is everybody okay with doing 2 o'clock like we did? Although I could pull it back to 1:15, but anybody who is under the assumption it starts at 2 will be late. Is the only concern.
Yeah, no, 2 is fine for me for Thursday.
We can do 2 this Thursday.
Okay.
Yeah, me too.
Okay, great. So we'll do 2 Thursday, and then we'll plan to meet every Tuesday, Thursday at 1:15. And then we can talk about on Thursday whether we want to plan for a meeting the following week, which is like during break.
For Thursday, how long will the Zoom like how long is the lecture going to be? Because I have another Zoom class from 2:30.
You have another Zoom class at 2:30, so they're meeting at a time separate from when you would ordinarily meet. Or
No, it's the same time.
Oh, that's after ours, right? Okay, so let me go back. You have a meeting on Thursday at 2:30?
Yeah.
Are you meeting every Thursday at 2:30?
Mm-hmm.
Yes?
Yes.
Okay. Well, if we start at 1:15 after this Thursday, then that shouldn't be a big problem, other than you're going to have 2 back-to-back Zooms. All right?
Yeah, no problem.
Okay. So I will tape this, Vera, so whatever you don't get to hear, you could go back into the Blackboard and watch the tape.
All
The
right.
best I can offer you, I think, for Thursday.
All right.
I'm sorry.
It's okay.
Okay. So then you guys need to, start reading chapter 10. And I'll tell you right now, it's a difficult chapter to plow through. Use the PowerPoints to guide you, because there are sections of that chapter we will not talk about. So my suggestion, look at the PowerPoint, start reading the chapter. On Thursday, we'll kind of talk about that. We can certainly try to go through the process as if we were doing a lecture, and see how that goes. How's that sound?
Sounds good.
Good.
Okay, any questions you want to ask about today's stuff?
No.
Okay. Well, then I guess we'll plan on seeing each other Thursday at 2. I will upload this recording, put it on Blackboard, and I'll see you Thursday.
All right.
Thank you.
Okay, guys.
Bye.
Thank you.
See ya.
Bye, everyone.
See you later.
Bye.
Bye.
Okay.